Hi guys, my name is Lydia Hunt, and for this lecture I'm going to be explaining a history of embryonic development and its modern implications. To begin, I will introduce my research article titled "Balls of Cells Mimic an Unseen Stage of Human Embryo Development." This article details studies conducted on human stem cells that have unlocked a new understanding of the earliest mechanisms of embryonic development, further detailing how this may relate to understanding the development of human disease, miscarriages, and birth defects. The duration of this lecture will focus on how embryonic development has been understood throughout history and how this understanding has led to advancements such as the one mentioned in this article. I will continue this discussion by introducing a history of embryonic development, beginning with early understandings of it. We will start with the theory of epigenesis. It was a theory proposed by Aristotle in an effort to explain development in sexually reproducing organisms. By examining the hatching of chicks, he was able to formulate his theory. What he observed was the fact that there were material, final, formal, and efficient causes at work in developing individual organisms. The early egg was not formed, nor was a miniature version of an already formed chick. Instead, the organisms gradually obtained its form. These observations led him to formulate the theory that when the fluids of a mother and father organism come together during reproduction, it sets in motion the mechanism of development and contains all of the necessary contents to give rise to a new organism. With epigenesis came the concept of vitalism, that withheld that there is a vital force operating in the living organism and that this cannot be reduced or explained simply by physical or chemical factors. The theory of epigenesis was contradicted by the theory of preformation, which began in materialism, a Cartesian philosophy that the world is mechanistic and deterministic, absent of any vital force driving or interfering with interactions of physical objects. The theory of preformation arose from materialism and suggested that all living organisms existed as a whole prior to their development and consisted of preformed parts. More specifically, they existed as miniature versions of themselves. Preformation rose to predominance in the late 17th century and withheld until the late 18th century. Its downfall occurred on account of the fact that the introduction of cell theory and better microscopic technology allowed scientists to view the very earliest stages of embryonic development in more detail. What they saw was bundles of cells and not preformed humans. We will continue this discussion by laying the foundation for the field of experimental embryology, beginning with Wilhelm His. His invention of the microtome in 1866 allowed him to create more thin and uniform cross-sections of preserved embryos than had ever been seen before. This provided better quality, more complete specimens that improved the ability to understand how developmental processes shaped the individual. Next is Eduard Pflüger. His experiments with frog embryos highlighted the idea that development was not a strictly internal affair, and that manipulations of environmental factors could give rise to changes in the developmental pathway of an organism. This claim was made clear in his tests of the effects of gravity on the orientation of development. He believed that this revealed that changes in gravitational field caused changes in location of the first cleavage plane in developing embryos. Finally, we have Wilhelm Roux. His developmental mechanics suggested that experimentation is the proper causal method of investigation. His half-embryo experiments led him to believe that development occurs internally from the nucleus and follows a predetermined mosaic. Roux's work pioneered a shift from descriptive to experimental studies of embryonic development. The next step historically for understanding embryonic development was understanding organization during development. This was achieved through Hilde Mangold and Hans Spamann's organizer experiments of 1921. Experimental proof from Mangold's salamanders and Spamann's hypothesis facilitated the discovery of specific organizer regions in developing embryos. In amphibian embryos, a cluster of cells gives rise to the development of the central nervous system. Their experiments led to the discovery of the concept of induction, which remains an integral doctrine of the developmental biology field. It withholds that the identity of certain cells influences the developmental fate of its surrounding cells. Once it was understood how embryos developed, it was only natural to question what went wrong during development. Enter teratology. The natural place to start seems like Meckel's monsters. Cataloging embryos with monstrosities allowed Johann Friedrich Meckel to develop a range of normal developmental patterns in a handful of organisms and paved the way for the emergence of teratology. Malformation was not unnatural and resulted in deviation from normal developmental patterns. One such defect discovered by Meckel was Meckel's diverticulum. In this condition, the connection between the fetus's intestine and the mother's yolk sac does not close, resulting in internal bleeding in the newborn. Meckel linked this abnormality to a problem that occurred during embryonic development. In his theories of malformations, he identified primary and secondary malformations. A primary malformation was a developmental abnormality, while a secondary malformation occurred due to a previously existing malformation. Continuing this discussion, we will address James Grave Wilson's 6 Principles of Teratology, published in 1959. They guided research on teratogenic agents and their effects on developing organisms. They relate to genetic susceptibility, developmental stage, mechanisms, endpoints, access, and dose response. Wilson stated that teratogenesis was the destruction of a critical number of cells in excess of that which the fetus could restore by later proliferation, thus prohibiting normal development. He also believed that these effects may occur at any point post-conception but prior to parturition. Now that we understand a history of developmental embryology, let's revisit the research article and its experimental findings. Gastrulation is a process that occurs approximately 14 days after conception when the embryo elongates, forms layers, and reveals a rough plan for the body. Prior to this study utilizing bundles of human stem cells, this stage of development had never before been seen in the lab due to ethical and technical constraints of using surplus human embryos from in vitro procedures or animal embryos. Structures made from stem cells, called gastruloids, have successfully mimicked some features of embryos post-gastrulation, which could reveal how genetic mutations and chemical exposures can lead to miscarriages and birth defects. Gastruloids lack the extra embryonic cells that would eventually implant the embryo in the uterus and form the placenta, as well as the precursors to what would become the embryo's brain cells. Now that we have revisited the research article, it is important to consider the ethical implications of stem cell embryology. We will be beginning by explaining the 14-day rule. This states that a human embryo may be studied experimentally no longer than 14 days, which is approximately when the first semblance of the nervous system appears. Comparing gastruloids, as pictured on the right, to human embryos could inspire changes to this rule. In addition, researchers could be inspired to add extra embryonic cells and brain cell precursors to these gastruloids in an attempt to model the entire human developmental cycle in vitro. Ethical ambiguity could result in pushbacks against the 14-day rule, as well as difficulty regulating what can humanely be done to these modeled embryos. Finally, let's consider stem cell embryology in the future. Cambridge developmental biologist Naomi Morris, co-author of the gastruloid study, says that in the future she plans to use these embryonic models to study how cells self-organize. Goals of the study moving forward are to introduce mutations to the stem cells that form the gastruloids, as well as to form reprogrammed units using cells from individuals with congenital disorders. This will help uncover how developmental processes can go wrong and lead to birth defects or sudden loss of pregnancy. The goal of this study is to build upon what scientists like Heath, Roux, Meckel, Spemann, and others have been trying to understand for centuries, and will hopefully lead to a better understanding of developmental defects, treatments, and preventative measures for the future.